Hello everyone, this is the introductory video for lab this week. We're going to be discussing animal nutrition and some of the chemical and physical processes that occur when you consume food. You should make sure to read through your lab handout that should be posted on BIOL213 before you come to class to familiar yourself a little bit with what we're doing in lab this week. You should also become pretty familiar with this figure that's shown on this slide here. And this is just showing the different substrates that we are going to look at as far as the digestion of these different substrates through the enzymes produced in different parts of the body. And so we're going to look at the digestion of carbohydrates, proteins, and fats that are all found in our food, and how the enzymes that are produced in different parts of our body break apart those substrates into end products, such as sucrose is the end product from the breakdown of starch. We get the end product of amino acids from the breakdown of protein digestion. And we get, these monoglycerides and fatty acids from the breakdown of fat. And the breakdown is facilitated by these different enzymes that are found that are produced in different parts of our body. So just make sure that you're really familiar with this figure, because you're going to refer to it quite a bit as you move through the lab, and you're going to use it to answer some of your questions and on your question handout. And so before I talk about some of the activities, these are some of the objectives for the lab. So you should be able to list digestive enzymes involved in digestion of proteins, lipids, and carbohydrates, and know the end products from the digestion of these substrates. Should be able to define enzyme, catalyst, the different substrates. Should know what hydrolase is and what an experimental control is. And you should also be able to describe the different assays that we used, specifically because we're going to be writing a method section. And we'll talk a little bit about that more towards the end of this video. And you should be able to determine if a particular substrate's been digested from the results of the assay. And you'll need to discuss the role of pH, discuss the role of pH and temperature on enzyme activity. And you should know the function of bile and its role in fat and oil digestion. And then some of the physical processes that we're going to look into today is process of swallowing. You should be able to talk in detail about how swallowing is voluntary action at first, and then as the process proceeds, it becomes this more involuntary action. And so you should be able to talk about swallowing in detail. And then also you need to be able to compare and contrast the different mechanisms for moving food through the digestive tracts, but also mixing food in the digestive tracts. So you should be able to talk about, there's two different mechanisms that you should be able to compare and contrast. And so these are activities 1 through 3, and these are the breakdown. These are, where we're looking at the breakdown of these different substrates through these different experiments and running different assays on each of these substrates. So we're going to break up into groups of 3, and you're going to assign one person per experiment. So one person's going to work on activity 1, another person activity 2, and third person on activity 3. And then as you're moving through these experiments, make sure these experiments and assays, make sure you're recording them on your results on your questions handout, and also answering the questions for each experiment. And so activity 1, we're going to look at the digestion of starch through amylase, salivary amylase produced in the mouth, and record the try to quantify, but just look at the presence of the end product at the end, basically, so maltose for the breakdown of starch. Our second activity is going to be digestion of protein. We're going to be using this BAPNA, and essentially this is just attached to the protein, and when trypsin when we add this enzyme trypsin to the protein, it essentially creates this color change, and then we're able to tell if it was digested, and then the end product for protein digestion are these small peptides. And then activity 3 is the digestion of fats and oils, and it's a little bit, as you can see, a little bit more involved chemical process, chemical process compared to the first 2 substrates, so you should be able to talk about bile in general and how it's used in the breakdown of fats and oils. And we're going to be looking at lipase as the main enzyme for breaking down fats and oils, and how and then looking at the byproduct, which is monoglycerides and fatty acids. And so make sure you're recording your results as you move through those as a group, but we're also going to be recording and keeping track of people's each group's results, and sharing them with the class. So your TA should come up with some mechanism for each group to record their predictions and results, so that they can share them, so each group can share them with the rest of the class. And so the 5th activity, we're not actually going to be doing this semester in lab this week, because if you'll read through the lab handout for activity 5, it involves, we generally have students swallow water, and then you're sort of tracking the process of swallowing. And because of COVID this year, we just don't want students to be pulling down their masks during lab. So we're actually going to not be doing this activity this week. However, you should still certainly understand everything from activity 5. You should be reading through it and making sure you're understanding the process of swallowing, and be able to talk about how it's this voluntary action at first, and then it becomes this sort of involuntary action as the process proceeds. And so you can work on this, read through this activity 5 as you're going through some of these the first 3 activities, because you're going to have some downtime, and so that's a great time to read through activity 5 and make sure you're understanding everything in that section. And then activity 6, we're actually going to show a video in class to look at both segmental and peristaltic movement, and essentially looking at, you should be able to compare and contrast these different movements, peristaltic movement, segmental movement, where peristaltic movement is essentially a process that moves food through the digestive tract, where segmental movements are more involved in mixing of food by using digestive juices. So you should be able to talk a little bit about how digested food is moved through the digestive tract, through these either moved or mixed through the digestive tract through these different processes. And then last thing, as I mentioned earlier, is we're going to continue on practicing becoming excellent scientific writers. And so that means this week we have another writing assignment. But this week we're going to just focus on the material and methods. And so you're only going to focus on the material and methods from activities 1 through 3. So the digestion of starch, proteins, and fats, and just some general notes for points that may be taken off if you're missing some of these components. It needs to be typed and double spaced. You need to write in first person. Ensure that your ideas are well organized and easy to read. You should definitely proofread it before you turn in the assignment. Look for any grammatical or syntax errors and make sure those are corrected before you turn it in. And then this is going to be our material and method section, so make sure to label your section. So the materials and methods should only be rough, 1 to 2 paragraphs, and should not be more than 1 page. Just recount the procedures you used as you ran through the experiments, and and basically describe things in a way that are concise. You know, you need to, if you handed you this material and method section to somebody that didn't go through this lab, they should be able to read it and replicate the experiments exactly. Even if they're coming up with different results, they should be able to go through the process of running through the experiment fairly easy and with little question. Make sure material and method sections written in paragraph form. Please no bullet points. And do not include your results. We're just focusing on the material and methods for this writing assignment. So don't include your results. And so like I had mentioned, this is going to be due a week from next week's lab. So you'll have, you know, a week to work on it, and just ask your TAs if you have any questions as you're working through it. And I think that's basically it. So hope everyone has a good lab this week and has a fun time. So.